Hi. In the last lecture, the characteristics of cells and viruses, we have discussed that all living organisms are made up of cells. Now, we will see that there are two basic cell types in nature, the prokaryotic and the eukaryotic cells. Prokaryotes are organisms that have prokaryotic cell, and eukaryotes are organisms that have eukaryotic cell structure. The study of prokaryotes has been divided into two parts. This lecture will focus on prokaryotes in part 1. Here is an overview of the study of prokaryotes part 1 and 2. We will first go over the characteristics of prokaryotes, the taxon, and examples of prokaryotes. Then we will look in detail the structure of prokaryotes, going from the outermost parts to the innermost parts of the prokaryotic cell. We will study the external structures, the cytoplasmic membrane, the cytoplasm, and the internal structures. In part 1, we will first go over the characteristics of prokaryotes, taxon, and examples of prokaryotes. Then we will look in detail the external structures of the prokaryotes, which are the capsule, shown here in orange, the pili and the fimbria, shown as blue lines all over the surface of the bacteria, the flagella, shown in dark orange, and the cell wall, shown in yellow. We are choosing the bacteria as the main representative of the prokaryotic cell. But first, let us look at the characteristics of prokaryotes. By definition, the word pro means before and karyote means nucleus. Prokaryotes are microorganisms that existed before there was a nucleus in terms of evolution. Prokaryotes therefore do not have a nuclear membrane surrounding their genetic material, that is the DNA. The region where the DNA is found is called the nucleoid. Prokaryotes also have no membrane-bound organelles in their cytoplasm, such as the mitochondria, or in other words, they lack a number of internal structures that are bound with a phospholipid membrane. The DNA, or the chromosome, of the prokaryotes is small and circular, double-stranded. Some prokaryotes also have plasmids, which are separate from the chromosomal DNA and replicate independently from the chromosome. Overall, prokaryotes are relatively small, around 1 micrometer or smaller in diameter, and simple microorganisms compared to eukaryotes. Now that we have studied the characteristics, let us move on to the taxon. Taxonomy is the science of classification of organisms, and a taxon is a category or a group of organisms that have shared characteristics. So, in the classification of organisms, prokaryotes are found in two main groups or domains, the domain Bacteria and the domain Archea. Archaea are known to predate bacteria in terms of evolution. The main difference between these two domains are in the type of lipids in the cytoplasmic membrane that are found in each of these domains, and in the chemistry of the cell walls, which we will look into detail later on. Shown here are a few examples of Bacteria and Archea. The first one is Streptococcus pneumonia, and the second Escherichia coli, the third Staphylococcus aureus are examples of bacteria. For Archaea, we have Sulfolobus, which is a hyperthermophile, meaning it survives in extremely high temperatures, and a Methanogen, which is a methane producing Archaea. Let us start with the capsule or the GLYCOCALYX. As the external structure. Other external structures that we will focus on are the Pili and Fimbria, the flagellum, and the cell wall. In each of these, I'll be showing images of the structures and going over the function and the types if needed, and also the differences between bacteria and archaea and between gram-negative and gram-positive bacteria. Let us start with the capsule or GLYCOCALYX. Glyco means sugar and calyx means cup. GLYCOCALYX is composed of polysaccharides, polypeptides, or both. The GLYCOCALYX is produced inside the cell and then extruded onto the surface of the cell. If the GLYCOCALYX is firmly attached, it is called a capsule. If it is loosely attached and if it tends to be water soluble, it is called a slime layer. GLYCOCALYX tends to be found in pathogenic bacteria, meaning bacteria capable of causing disease. Slime layers help with attachment to surfaces. An example of this is a bacteria that colonizes the oral cavity. Capsules, on the other hand, help prevent desiccation and help the bacteria in resisting engulfment by defensive cells, also known as the process of phagocytosis. Examples are Streptococcus pneumoniae and Klebsiella pneumoniae. Panel A, shown here, is a scanning electron microscopy of three kinds of bacteria, namely Streptococcus pneumoniae, Klebsiella pneumoniae, and Bacteroides fragilis. Panel B is a negative stain for capsules of Streptococcus pneumoniae and Klebsiella pneumoniae. So, what you see in dark blue is the bacteria itself inside the capsule, and so everything which is white and surrounding the bacteria is the capsule, shown by these blue arrows. The capsule is white because the dye cannot penetrate the thick polysaccharide layer of the capsule. Because of the looseness of the slime layer is easily washed off and thus cannot be stained in the case of Bacteroides fragilis. Finally, shown in the bottom panel, the panel C, are the transmission electron microscopy images for both the capsules of Streptococcus pneumoniae and Klebsiella pneumoniae and the slime layer of Bacteroides fragilis. Fimbria and Pili, shown here as an example, are external rod-like projections made up of protein. They extend several micrometers from the cell surface. The main functions are adherence, conjugation, and biofilms. It is now noteworthy to mention that Pili and Fimbria are used interchangeably by some scientists but not by others. Fimbria help with adherence to the environment. Some scientists name the Fimbria that help with adherence to another bacteria for the purpose of transfer of genetic material as conjugation pili or sex pili. Therefore, pili are also known as conjugation pili. The words therefore pili and Fimbria can be used interchangeably. However, pili tend to be longer than Fimbria, shorter than flagella, and tend to transfer the DNA from cell to cell. Example of a bacteria that has Fimbria is Neisseria gonorrhea. The bacteria has several Fimbrias that enable it to colonize the reproductive tract by attachment. Here, figure A is a representation of Fimbria. Figure B is an electron micrograph of E. coli showing Fimbria. C is showing sex pili as these long elongations, the other name for the conjugation pili, in contrast to the smaller Fimbria that are found all over the surface of the bacteria, and which are much more in number than the sex pili. In D, you can see both Fimbria and the conjugation pili again. The conjugation pilus literally pulls the other bacteria close, similar to pulling something with a rope in order to have the transfer of genetic material. Now, flagella are long structures that extend beyond the surface of a cell, including the GLYCOCALYX, and they propel the cell through its environment. The singular for flagella is flagellum. In terms of structure, flagella are made up of flagellin, which is a protein, the hook, shown here, and the basal body. In terms of its location, flagella can be found outside the cell, in which case it's an exoflagella in its main part, or the main part may be inside the cell, endoflagella, as in the case of spirochetes. In the periplasmic membrane, which gives the spirochete its curved shape. The function of the flagella is locomotion. It allows the bacteria to rotate 360 degrees. The movement is mainly by runs that are punctuated by tumbles. The runs occur when the rotation is counterclockwise, whereas the tumbles happen when the rotation is clockwise. Based on the type of flagella, bacteria can be classified as peritracheous, those that have the flagella uniformly distributed all around the body, monotracheous, only on one side, on one location, lophotracheous, several, and polar, meaning several on one side or the same pole, and amphitracheous, meaning a single flagellum located on each side of the pole. Therefore, this is amphitracheous and polar. In terms of its structure at the level of the shaft, prokaryotic flagella has 9 triplets of microtubules and no microtubules in the center, as shown in this electron microscopic picture at the bottom and in the representation on the top. 3 microtubules, triplets, 9 sets with none in the middle. Shown here are a gram-negative bacteria and a gram-positive bacteria. I will explain the difference between these two types of bacteria in the next few slides. For now, the main difference between the gram-negative and the gram-positive flagella is that the gram-negative flagella is more complex in that the basal body is more complex and is located across the inner membrane and the outer membrane of the gram-negative bacteria, whereas the basal body of the gram-positive bacteria is much simpler, located on the cell membrane. Bacterial cell walls are composed of peptidoglycan, also known as murine. Murine consists of N-acetylglucosamine, also known as NAG, and N-acetylmuramic acid, also known as NAM. In terms of function, cell wall helps in giving structure and shape to the bacteria or the prokaryote. It also helps protect from osmotic forces and aids in attachment. The cell wall also helps in resistance against antimicrobial agents. Now let us look at the gram-positive and the gram-negative cell walls. These bacteria named as such, gram-positive and gram-negative, according to a differential staining technique known as gram staining, about which we will study at a later time. When comparing and contrasting the two, both gram-positive and gram-negative cell walls are similar in that they both contain the cytoplasm and they both have the cell membrane. Now, while the gram-positive bacteria has a thick peptidoglycan layer, the gram-negative bacteria has a very thin peptidoglycan layer. And the gram-negative bacteria has an outer membrane of lipopolysaccharides, while the gram-positive bacteria does not. Therefore, the gram-negative bacteria is double-membraned, whereas the gram-positive bacteria is single-membraned. Now, the archaeal cell walls differ from bacterial cell walls in that they do not contain peptidoglycan. Some archaeal cells may have what is known as pseudomurine, which literally means false murine. Pseudomurine is made up of N-acetylglucosamine and N-acetylthalassominuric acid. Other archaeal cells may have polysaccharides, proteins, or both. Some archaeal cells contain the S layer, which is a hexagonal pattern of protein and glycoprotein. The function of archaeal cell walls is mainly mechanical support and prevention of osmotic lysis. So, in summary, prokaryotes have no nucleus because they lack a nuclear membrane. They have no compartmentalization of function because they lack membranous organelles, and they have a circular DNA. The two main domains are bacteria and archaea. The external structures we went over were the capsule, which is found mainly in pathogenic bacteria, the Fimbria, which allows for attachment, and the Pili, which allows for conjugation, the flagella, which helps with locomotion, and the cell wall, which is medically important due to the resistance against antimicrobials. This concludes part 1.